In Chapter 15, we are going to examine the sensory pathways in the somatic nervous system. So just to remind you, the afferent division of the nervous system is the sensory division. So we're going to have our receptors that are going to be distributed all over the periphery, and the information that they collect from the environment will be conveyed via sensory neurons to the central nervous system. So our sensory afferent division is going to converge on the central nervous system, and we're going to have our sensory neurons. If you recall, they have their neuron cell bodies in those ganglia of the dorsal root, so within the dorsal root ganglion. And then those neurons will synapse somewhere in our somatic sensory nuclei in the posterior gray horns of the spinal cord or in brainstem nuclei. And from there, they will ascend in the central nervous system to the brain, either in some processing centers in the brainstem or possibly up to the thalamus, where they may then reach the cerebral cortex. Now, our efferent division is going to be the motor division. And so we're going to have our motor neurons originating in the brain, and they will travel via tracts within the central nervous system, either to brainstem nuclei or to motor nuclei in those anterior gray horns of the spinal cord. And then they will synapse in motor nuclei. These will be our upper motor neurons. And they will synapse on lower motor neurons within those nuclei. And those will be our efferents that exit the central nervous system and will target effectors. All right, so let's look at the sensory pathways. That's everything in this purple. So when we have a sensory pathway, the first thing that's going to happen is we're going to have some kind of stimulus arrive at a receptor. And so this is going to produce some kind of potential in the membrane of a receptor cell. And that can generate an action potential downstream. So if we reach threshold, either the receptor itself is a neuron or the receptor will be coupled to a neuron. And if we can bring that neuron to threshold, then we will generate an action potential, and this will generate sensation. So sensation is sensory information arriving at the central nervous system. That sensory information could be used one of two ways. We could have an immediate involuntary response that's mediated through reflexes. So for example, the withdrawal from the hot stove reflex. And this happens before we're even consciously aware of what's going on. And that's a protective reflex. That's to keep us from burning ourselves. Or we could also have that information conveyed up to higher areas of the brain. So some of that information will get relayed to the thalamus, and the thalamus will filter it out and decide whether it needs to be conveyed on up to the primary somatosensory cortex or the primary sensory cortices of different areas. For example, the occipital area, we know the occipital pole is our primary visual area, and our superior temporal gyrus of our temporal lobe is going to be our primary auditory area. Anyway, these sensations may reach their relevant areas of cortex. Right now, we're going to be focusing on somatic sensation, so we're going to be talking about sensation that's going to be reaching that post-central gyrus, or basically the primary somatosensory cortex. And if it reaches the conscious awareness and the thalamus lets it through and reaches our conscious awareness, then we can use that information. We become consciously aware of it, and we not only sense it, but at this point we perceive it. So in order for us to perceive a stimulus, it must reach cerebral cortex. Sensing a stimulus is essentially turning on a receptor that will then send a signal down a sensory neuron that will reach the central nervous system. So this is sensation. But perception is if that sensation gets relayed up to our primary cortex, so our primary sensory cortices in our cerebral cortex. All right, so sensory receptors are going to be specialized cells, and they're going to be receptive to specific things like touch, pain, temperature, vision, hearing, and these kinds of things. And we'll see that there are two different main types. We're going to have general sensation, which are distributed all over the body, and special senses, which are things like vision and hearing. And we'll talk about those in more detail a little bit later on. But our sensory pathways, as we know, are the afferent pathways that will ascend from where they synapse in the central nervous system up to higher brain areas. So in the periphery, we're going to have nerves, and we're going to have axons traveling within those nerves through the dorsal roots, and they will have their cell bodies in the dorsal root ganglia, and they will synapse in those posterior gray horns in the somatic sensory nuclei. In some cases, we'll have some of them synapsing in brainstem nuclei. And from there, we're going to have tracts, white matter tracts, that will then convey that information through the central nervous system to higher brain areas. All right, so we know that our somatic motor commands are going to travel from motor centers in the brain along the somatic motor pathway via these motor nuclei in the spinal cord and the brainstem. So now here we're going the opposite direction. So we have the axons traveling in with the central nervous system will be descending in tracts that will then, those neurons will then synapse in nuclei, usually within those anterior gray horns of the spinal cord or perhaps in some brainstem nucleus. And then the lower motor neurons will exit as the efferent via nerves. So once we get out of the central nervous system and into the peripheral nervous system, we call these collections of axons, these myelinated axons, nerves. So our somatic nervous system is going to be the motor neurons and pathways that control skeletal muscles. But in this chapter, we're also going to be talking about our somatic sensation. So this is going to be all the information that's gathered on the periphery via peripheral receptors and information about our external environment that's going to be brought into the central nervous system. Now, we're going to focus on this one on our somatic sensation, which are basically our general senses. Things like pain, touch, temperature, pressure, vibration, and proprioception. And proprioception is where our joints are in space. So our receptors for this will be muscle spindles, free nerve endings, and joints, and things like this. All right, so as we've said before, sensation is simply the arriving of information in the central nervous system from the sense that we're monitoring. Perception is the conscious awareness of that sensation. So it has to reach the cerebral cortex in order to become a perception. Otherwise, it's just a sensation. We get many sensations bombarding us every second. We're getting sound, visual, tactile sensations all the time that we're not aware of because it would just simply overload our conscious awareness, so the thalamus filters them out. So perception is the conscious awareness of a sensation, and this is a key thing to get because a lot of people miss this on the test. Perception is conscious awareness of a sensation, and it has to be relayed all the way to cerebral cortex. If a sensation does not get relayed to cerebral cortex, it will not become a perception. Your body may act on it subconsciously, but you may never become consciously aware of it. And we will see there's a lot of sensory information that is carried to areas of the brain like the cerebellum, and the cerebellum uses that information to adjust ongoing postural control and motor movements. But we never become consciously aware of that sensation because it doesn't get all the way to our cerebral cortex. All right, so we talked a little bit earlier about what our somatic senses are. Now I'm going to touch briefly on the special senses, and we have a whole chapter dedicated to them later on. And these are special senses that are located in special sense organs in the head. The receptors are typically quite a bit more complicated than the ones that we will see in the general sensation. Plus, they're not distributed all over the body. They're in a very specific organ. For example, olfaction is going to be in the nose, vision is going to be in the eyes, gustation is going to be in our taste receptors, our taste buds on our tongue. Equilibrium and hearing will both be contained within the ears. So these are very specific to specific modalities. So vision is light. Hearing is going to be sound pressure waves in the air. Olfaction are chemical odorants. Gustation are taste chemicals that are going to be coming from food. And equilibrium will be the way that certain sensory receptors in our ears are going to interact with gravity and inertial changes. So all of these are special senses, and to hit home this point, special senses are all located in the head. They all have a specific special sense organ, and their receptors are quite a bit more complicated than the general senses, which are distributed all over the body. And these are simple things like pain, touch, temperature, pressure, vibration, and proprioception. Now, as we've said, the special senses, we've already talked about these, where they are, but all senses are going to have a receptor specificity. And that means that it's going to be sensitive to certain things. So touch receptors will be sensitive to touch, and that touch might be light touch, it might be pressure, it might be vibration. All of those are types of touch, and we have some touch receptors that are more sensitive to pressure than, say, others, and some that are sensitive to light touch. And we'll talk about those a little bit later on, but they're all sensitive to touch. We will have thermoreceptors that are sensitive to temperature, so heat and cold. And we will have some receptors when we look at our special senses. For example, the eyes are specialized for the detection of light. They're not going to be responsive typically to pressure. And if they do respond to pressure, it's going to transduce or turn that signal into a neural impulse, but they are going to transduce that signal as a light stimulus. So if you've ever rubbed your eyes and seen lights, that's because your eyes are specific for the detection of light, and anything that stimulates them is going to be interpreted as light. And each receptor is going to have its own receptive field, and that means a specific area that it will monitor. So any stimulus occurring outside of the receptive field is not going to be sensed by that receptor. So let's have a look. Here we have two touch receptors in the skin, and we have one, and its receptive field is going to be the extent of, these are free nerve endings, so this is going to be the extent over which these free nerve endings are distributed in the skin. And receptive field 2 over here is going to be associated with this receptor, and we've got these free nerve endings distributed through the skin here as well. But we will see that the extent of this ability for this receptor to detect something, well, it has to fall within its receptive field. So if we had a stimulus, let's say we poked with a needle or something, and we poked here, then we would be within this receptive field, receptor number 2 here. And so we would stimulate this receptor. Receptive field 1 was not stimulated, so receptor 1 is not going to convey any sense of the stimulus, only receptor 2. If we stimulated right here, and there's not an overlap of the receptive fields, or they're not touching, then we're not going to feel anything at all, because the stimulus did not fall within the receptive field of any receptor. And that's an important point, because if we stimulate outside of a receptive field, that receptor will not sense the stimulus. And if we stimulate where there is no receptive field, we're not going to feel anything at all. All right, so each type of sensory information is going to be brought by what we call a labeled line. That means that it's going to travel on specific pathways, specific tracks to specific areas of the brain to be interpreted. So for example, light will be detected by the eye. It will travel via visual pathways through the thalamus, through that lateral geniculate nucleus, and it will end up in our occipital cortex and our primary visual cortex. And so that would be the labeled line, and that would be the pathway by which it would travel. And sound would travel by different pathways, so it would travel from its receptors in the ears into the various brainstem areas that are going to process that sound, up to the medial geniculate nucleus of the thalamus and into the superior temporal gyrus. So we've got different pathways that are associated with different senses. And likewise, when we think about these somatic senses that are distributed all over the body, they are going to reach that primary somatosensory cortex, which is in the post-central gyrus right behind the central sulcus. And we will find that actually, that whole cortex contains a map of the body. So it's going to tell us where these sensations came from. So a sensation from our finger is going to innervate a different place in our primary somatosensory cortex than if we stimulate the foot or the knee or the head. So we're going to contain a map of the body within the primary somatosensory cortex that's going to tell us where these sensations are coming from. And one of the things that's important here, once again, to note is that if you stimulate outside a receptive field, you're not going to bring that information to the cortex, because you're never going to have stimulated a receptor at all. So even sensation will not happen. All right, so let's talk about an interesting phenomenon called synesthesia. Synesthesia is where we mix up some of these labeled lines. So sometimes you can have some sound or light or touch be interpreted as a completely different modality. And this could happen at a variety of steps along the way. It could be the actual labeled line gets mixed up. It could happen in the sensory interpretation areas. But sometimes we will find that a particular stimulus will be interpreted as something else. And to give you a concrete example, there was a very famous musical composer by the name of Rimsky-Korsakov, and he had what we call synesthesia. So anytime you played a specific tone or note, he would hear a color associated with it. So he would have a color association with each note in the scale. And this is an interpretation that never diminished any time in his life, nor did it ever change. So these color maps that were associated with tones, auditory tones, stayed with him throughout his entire life, and they were indelible. And so this is some kind of sensory processing mix-up, and sensory synesthesias are actually not that uncommon. That's just a very striking example of one, and there are people even today who have these sensory synesthesias where they'll actually hear colors. Now let's talk about a property of perception and even sensation called adaptation. And this is where you can shut down irrelevant stimuli. And this can happen in one of two locations. It can happen at the receptor itself, so you can shut down the receptor's response to a stimulus, so you're literally shutting down sensation. Or you can have central adaptation, which occurs in the central nervous system, where you're still receiving the sensations, they're coming into the central nervous system, but the central nervous system is shutting them out. So in either case, basically, you're ignoring stimuli that are irrelevant, that are either not noxious, not harmful, and they don't require your immediate attention. So things like the projector in the classroom, or the hum of the fluorescent lights, or the sound of the air conditioning, or any of these number of things are things that our body tunes out because they're irrelevant. And once again, they would just overload our conscious perception of these sensory stimuli. So we have two ways to adapt. We have peripheral adaptation, literally going to shut down the sensation by shutting down the receptor. And then we have central adaptation, which is going to shut down the central nervous system's response to that sensation, and it's basically going to prevent it from conveying that sensation up to cortex, where it will then be perceived. Let's look at some of the mechanisms of peripheral adaptation. So we've got two types of receptors, generally speaking. We have some that are called tonic, and these are always active. That means there's some kind of baseline firing rate where they're firing action potentials, and then if you stimulate them, they will increase their firing rate. And they will continue to have an increased firing rate as long as you stimulate them. And then when you stop stimulating them, then their firing rate will fall back to baseline. And we call these slow adapting because they typically don't ever stop firing. And the sensation is never shut down. They're always carrying some kind of sensory information. A good example are certain types of pain receptors. So we have a type of pain that lingers on after an injury, and this is usually that burning, sort of poorly generalized pain, and it's usually carried on unmyelinated fibers, and it's just this irritation around the area of injury. And this is an example of slow adapting receptors, or tonic receptors. They're always firing. Here's an example of how they work. So here we have our baseline firing. Here's an action potential, and there's no stimulation, say. So now we stimulate, or we increase the stimulation, and they will increase their firing rate. So a lot of times, let's just say a sensory receptor in the skin for touch. Well, usually, for example, the shirt on my body here is touching me and it's causing sensation. And there are certain types of receptors that we will see will basically shut down. And then there's some that will continue to convey that sensation. And we'll look at different touch receptors that respond in this way. We'll see that there are both tonic and what we call phasic receptors, which are fast adapting. And we will see that these receptors, the tonic receptors, are going to be firing no matter what. If they're being stimulated, they're going to fire. If you increase the stimulus intensity, they're going to fire more. If you decrease the stimulus intensity, then they'll reduce their firing back to baseline. And they will always generate action potentials. These action potentials may later be filtered out at the level of the central nervous system and be adapted centrally, but they're not going to adapt peripherally. So the receptor is not going to be turned off. On the other hand, we've got our phasic receptors that are fast adapting, and these are the ones that are going to become active only when there's a change in the stimulus. So it's going to provide information about an intensity or a rate of change of a stimulus. So they will only fire when something happens. So they can have a baseline amount of stimulation on them. Again, the shirt is stimulating my skin. And then there's some receptors that, unless something changes, they're not going to fire any action potentials. But now if there's an increase in, say, pressure or something, they might fire an action potential as that area is being compressed under that area of skin. And then once it's let go, then it'll fire again to let me know there's a change. So these are phasic receptors. They're normally inactive, but will become active when a stimulus changes. So this plays into this idea of peripheral adaptation. So our tonic receptors are slowly adapting. They're the ones that don't shut down. Our phasic receptors are the ones that are fast adapting, and these will pretty much shut down the receptors if nothing's stimulating them, or if the stimulus doesn't change. So that's the important point. If the stimulus doesn't change, then the phasic receptors aren't going to fire. Now let's talk about a very specific example of central adaptation. So if we look at olfactory receptors, they're tonic, so they're always active. So whenever there is the presence of odorant molecules in the nose and they're binding with receptors and those receptors will transduce a signal, that becomes a sensation. And that sensation will be relayed to the brain. And typically we will become aware of new smells as soon as we encounter them, because we will have a very strong sensation, and after a while though our brain will start to shut it down. So if the smell isn't changing and if the smell doesn't get worse or less, then we will become unaware of it after a while, although we're still sensing it. So our olfactory receptors are still transducing that signal and still sending the signal to the central nervous system, but our brain is shutting it out because it's irrelevant. So unless there's a change in that smell, then you're not going to be aware of it anymore. So a good example is when we're dissecting things in class and we pull out whatever it is we're dissecting, and it usually smells really bad. It smells strongly of formalin and these kinds of things. And most people find formalin irritating and annoying, and they'll smell it very strongly when they first come in the room. And even while we're sitting there doing the dissections, people will gradually become unaware of the smell, even though the concentrations haven't changed. And so after a while, people will leave the room and they'll come back in and they'll smell it very strongly again. This is a very good example of central adaptation, because while the receptors in the olfactory epithelium are tonic and always active and always conveying that sensation, it's shut down at the level of the central nervous system. So it's basically not reaching our conscious awareness. All right, let's talk about the classes of sensory receptors. And we can classify these in several ways, one of which is by their location. So when we talk about somatic sensory receptors, they can be located on the skin and the superficial areas, and these are going to be sensitive to the external environment, things in the external environment. We can have proprioceptors, and these are things like our muscle spindles. These are going to be receptors in muscles, tendons, joints, these kinds of things. And these are going to give us information about what our body parts are doing and where they are in space. Interoreceptors are going to monitor the functions of visceral organs. So we are going to feel distension of the stomach or of the bladder or other sensations coming from viscera from our interoreceptors. Now we can also talk about the types of stimuli that these receptors are going to respond to. For example, nociceptors will detect pain. Thermoreceptors will detect temperature. Mechanoreceptors will detect physical distortion of a membrane. So for example, if you remember back when we talked about our different types of channels, we have voltage gated channels, ligand gated channels, we had mechanically gated channels. And these are channels that, let's say, are sitting in a membrane somewhere, and when you physically distort that membrane, then the channel's open. So mechanoreceptors often have these mechanically gated channels and are receptive to physical distortion. Then we have chemoreceptors, and chemoreceptors are sensitive to changes in chemical concentrations. We will have chemoreceptors in the general senses that we never become aware of. These will be sensitive to things like dissolved carbon dioxide concentrations. These will be sensitive to things like hydrogen ion concentrations, and will help our autonomic nervous system regulate things like breathing, rate and urinary function and these kinds of things. So it's going to help us regulate the balance of fluids in our body and the balance of carbon dioxide and oxygen and these kinds of things. So we will see that chemoreceptors are something that in our general senses we never become aware of. However, we have two special senses that are also mediated through chemoreceptors. And when we think about it, we think about olfaction, which is basically binding with chemical odorants in the air. And those chemical odorants are going to activate the receptors. And gustation. So once again, we have chemicals in food that will be binding to receptors in the tongue. So chemoreceptors in general sensation are things we're not going to become aware of. Chemoreceptors in special senses are going to be our smell and our taste. Let's look at nociceptors first. We find them all over the place. We find them in the superficial portions of the skin, in joint capsules. If you dislocate your finger, you'll find that it really hurts because those joint capsules get stretched, and that's going to activate pain receptors. Also within the periostea of bone. So if you break a bone and you basically break through the periostea, then you could be causing tissue damage that's going to excite some of these pain receptors, and also around the walls of blood vessels. So interestingly, migraine headaches are caused by either excess vasodilation or excess vasoconstriction, and this is going to stimulate pain receptors within the blood vessel walls. So we have nociceptors that can be sensitive to temperature extremes. So sometimes the function of nociceptors and thermoreceptors overlap in that nociceptors will detect extremely high temperatures that are likely to cause tissue damage, things that will burn tissues, or really low temperatures that will cause tissue damage by, say, frostbite or things like that. They'll also be sensitive to mechanical damage, so excess stress, stretching, maybe tearing, ripping, these kinds of things, and dissolved chemicals. Often the chemicals released by the cells that were just damaged. So these will also excite pain receptors. And sometimes we'll have pain receptors that are sensitive to other things like acids or bases or caustic things that might get on you. So a lot of times these are free nerve endings, and they'll have lots of fairly large receptive fields. And we're going to have two types of pain. We're going to have fast pain, which is the fast prickling pain, and that will be carried on myelinated axons, type A myelinated axons, so larger diameter myelinated axons. So we'll become aware of this pain fairly quickly. And this can alert us to the fact that there's a painful stimulus there or something that caused that pain. But then we have our slow pain, which is mediated on these type C, sometimes A delta fibers, but either really, really thin, lightly myelinated axons or unmyelinated axons. And this is that kind of pain that just continues on, the burning, itching, kind of you don't know where it's coming from pain. And this is the kind of pain that reminds us to be careful of that area so that we don't damage it again. So we've already talked about the myelinated fibers and fast pain versus the type C fibers carrying the slow pain, so I'm not going to hover on these slides very long. Then we have our thermoreceptors. These are going to be sensitive to temperature changes. And we'll find them in the skin, within the skeletal muscles, within the liver, and of course within that preoptic area of the hypothalamus, because that's going to be our control center. That's going to be our thermostat that's going to regulate our body temperature. So these are going to be carrying temperature along some of the same pathways that carry pain sensations. And of course, both pain and temperature can arouse us because obviously we want to avoid pain. And for example, if we know what happens when we jump into a swimming pool and it feels really cold at first, that's going to cause a general arousal. So we're going to have a lot of these sensations sent to the reticular formation, not surprisingly. And as we remember, the reticular formation is going to regulate our state of awareness or arousal, this kind of thing. So also some of these will be sent to the thalamus and some will be relayed to the primary sensory cortex. All right, mechanoreceptors are going to be important for a lot of our touch functions. So these are going to be sensitive because they're going to have mechanically gated ion channels that will open when that membrane is distorted. So stretching, compression, twisting, other things. We will have, of our mechanoreceptors, we'll have tactile receptors. And these will sense touch, pressure, some vibration. And we'll look at several of these that we'll find in the skin later on. We're going to have baroreceptors, and these are pressure changes. And these are going to be specifically pressure changes, for example, in distensible organs like the walls of blood vessels, in the digestive tract, respiratory tract, and the urinary tract. So we're going to be able to feel when there's filling up a fluid, say in the bladder, this kind of thing. And we're also going to have proprioceptors. And the proprioceptors, once again, will be things like muscle spindles, Golgi tendon apparatuses, and free nerve endings that are in the joint capsules. Tactile receptors can be divided into fine touch and pressure receptors that are just sensitive to light touch and very light pressure, and crude touch and pressure. And so they're going to respond differently. We'll find that a lot of our fine touch will be tonically active, and some of our crude touch some of them will be phasically active, especially when we get into our pressure and vibrations and sensations. And we will find that our fine touch will be localized in areas like the fingers, the lips, areas that we're in contact with our environment. And these are going to be very sensitive, and they're going to have very, very small receptive fields. And we'll talk a little bit about why having small receptive fields is important when we move on to touch a little bit later on. But these are going to provide detailed information about our stimulation. So for example, if I pick up this phone, this case has all kinds of little knurling in it and tactile things like bumps and ridges to improve grip. So when I touch this, it's going to have some very, very different textures in it, but very subtle differences in texture. And you can feel these with our fine touch receptors. And so we're going to be able to when we hold something, we're going to be able to get information about its shape, its dimensions, its texture, and exactly where it is. So if I'm putting it on my finger, exactly what it's touching, and this kind of thing. Whereas crude touch and pressure receptors are going to have larger receptive fields. And one thing that's key to point out here is the smaller receptive field, the more of them you can pack together. So if you have an area of skin and you have tons and tons of receptive fields in it, then that means each one of those receptive fields is going to cover a very small area. So you can get a very good idea of the shape or dimensions or texture of something because you have so many receptive fields. And it's just like a photograph. So if you take a photograph with 20 megapixels, then you're going to have 20 million tiny little dots of light or tiny little dots of ink. And that's going to give you a much better picture than if you have a 10 megapixel picture in which the pixels are going to be larger. And so that means that the appearance will be grainier. So instead of having 20 million, you're going to have 10 million. And now you're going to have basically a much coarser picture. And the same is true with our receptive fields. The smaller and more densely packed the receptive fields, the better our sensation of that object, or the better that we can elucidate the features of that object. With crude touch, we might know that an object is there. We might know generally where it is, but we'll have much larger receptive fields. So if you imagine that I had receptive fields on my skin here and here, and I stimulate here, then this stimulation could occur anywhere in that receptive field. Whereas on my finger, if I have really, really tiny receptive fields, then I can differentiate what's in those receptive fields a lot better. So if I stimulate with this pen in one of these receptive fields here, then I can tell whether it was here or here. So I have a good ability to discriminate where I'm being touched with this pen because I have two separate receptive fields here that are relaying information about exactly where that pen touched. Over here, I could stimulate here or here or here or here and still not know exactly, just that it was somewhere in this area. Versus if I stimulate over here, I could stimulate in a lot of different areas here and only still be aware that it's somewhere in here and not have a really specific idea of exactly where in there it is. So the smaller the receptive field and the more densely packed they are, the better our sensation is going to be, or the more the better resolution our sensation is going to be. All right, so let's look at types of tactile receptors. First, we have free nerve endings. And here we see a bunch of free nerve endings that are distributed in the skin. So here they are a little bit in this, starting here in the dermis, at the papillary layer of the dermis, and extending up into the epidermis through our basilar layer into our spinous and granular layers. So these are just free nerve endings, and they're going to be sensitive to touch and pressure. And these are going to be tonic receptors. So these are going to be always active, and these are going to have very small receptive fields, and these are going to give us very distinct information about what it is we're touching. All right, next we're going to look at a root hair plexus. And this is just basically some dendrites wrapped around the hair follicle, so that if you disturb the hair within the follicle, it's going to twiggle the follicle around a little bit. And that's going to alert us that something might have landed on our skin, like maybe a bug or an insect or a mosquito or a spider or something, and we want to get rid of it. So it just alerts us to something that, might bite us or something like that. So we become aware of something that touches our hair that might then twiggle this hair follicle and stimulate this receptor. And this is going to be a phasic receptor. So this is going to adapt rapidly, and it's going to be really good at detecting quick stimuli like a landing of a mosquito on your skin and this kind of thing. All right, now let's look at the tactile discs. So the tactile discs basically really have two components. We have a Merkel cell, which is a specialized cell within the epidermis of the skin, and it's going to have, and it is a receptor in and of itself, and it is not a neuron, but it is a receptor, but it is going to be closely associated with a neuron with this very highly expanded dendrite. So this Merkel cell is going to excite this dendrite, and then we're going to have our afferent neurofiber here. This too is very sensitive to fine touch and in pressure. It's tonic, so it's always active, and it's going to have very, very small receptive fields, and this is going to give us a good sense of dimensions of objects and textures and that kind of thing. All right, so then we're going to have tactile corpuscles and lamellated corpuscles. And so let's have a look at them. It's easier to look at them. So the tactile corpuscles are in the dermal papillae, and they have this specialized sort of collagen capsule around them with these nerve endings within them. So these are going to be sensitive to pressure when you push down on the skin. So these are very fast adapting. So they're going to be sensitive to changes. So fine touch and pressure, low frequency vibration. So a very brief stimulus. Or if you have a vibration at a very low frequency on your skin. So this is the kind of thing that we're going to see tactile corpuscles are going to be responsive to. Then we're going to have lamellated corpuscles, which are the deep ones. And these are the ones that are also fast adapting. And they too are going to be sensitive to high frequency vibrating stimuli. So let's look at it. It's the next one. So here we have these layers, concentric layers of collagen that's going to surround this nerve ending, this dendrite here. And so basically that's why they're called lamellated, because the concentric layers are lamellae. And the more you push on it, the more you're going to deform this capsular layer, and you're going to stimulate this. So once again, this is a phasic receptor. It's very fast adapting, but that's why it's going to be very good for sensing vibration, because vibration is kind of like a sinusoidal vibration, just like that. All right, next we're going to look at Ruffinis corpuscles, and these are stretch receptors in the skin. So these are going to be tonic because they're going to let you know when your skin's being stretched. And these are basically going to be capsules of, or I should say collagen fibers within a capsule in the skin, and you're going to have a sensory nerve ending that is wrapped around this collagen fiber so that when the skin is stretched and the collagen fiber is stretched, then that's going to activate these sensory nerves. And this is going to show very little adaptation, and we're going to see a very similar apparatus in tendons. And we're going to see a Golgi tendon apparatus that has a very similar structure to Ruffinis corpuscles, except it'll be located deep, and it'll be located in the tendons. And basically, it will allow you to know that if your muscles are exerting too much force on the tendon. And so it'll basically signal your brain, and you'll have a nice reflex to to release the tension on the muscle so that you don't break the tendon. All right, now let's talk a little bit about our baroreceptors. These are typically rapidly adapting receptors, and these are going to monitor changes in pressure. So these are going to have nerve endings that are going to be within elastic tissues. We've already talked about where we'll find them in distensible organs such as blood vessels, the bladder, the lungs, the stomach, the intestine, things like this. All right, so proprioceptors we've already talked about, monitoring the position of joints. They will also monitor tension in tendons and ligaments, the state of muscular contraction, as we saw with our muscle spindles. And we will also find that these are for muscles and tendons and joints. We're not going to find proprioceptors in the visceral organs. So we're not going to tell where our spleen is or where our stomach is. We're not going to have proprioceptors that tell us where our organs are. So we're not going to be able to know, these things aren't moving around that much anyway, but we're not going to have a lot of ability to detect where these things are in the body cavities. All right, now let's look at the proprioceptors. We've already talked about the muscle spindles we talked about in previous lecture. Golgi tendons are very much like Ruffinis corpuscles. They work in a very similar way, except they're in the tendons. And then our joint capsules are going to have these free nerve endings that are going to detect stretch and changes in the joint capsule. So muscle spindles we've already talked about. They monitor the skeletal muscle length and are going to trigger the stretch reflexes. The Golgi tendon organs, basically located at the junction between the skeletal muscle and the tendon, and it is in the tendon itself, and it's going to be stimulated by tension within the tendon. So this is going to allow us to monitor how much external tension is developed during muscle contraction, and if for whatever reason that our muscles are about to overpower our tendons, we actually have a reflex that allows us to release our muscle tension so that we don't pull the tendon, say, right out of its insertion. And this is actually fairly common. People can override this reflex. So bodybuilders in particular are notorious for this, especially when they're doing their preacher curls, and maybe they use a little bit too much weight and they pull, and they're forcing their muscles to contract even when the tendons can no longer handle the load. And in a normal reflex, you would drop the weights. But sometimes these guys will overpower that reflex and they'll just pull the tendon right out of the insertion on the radial tuberosity. All right, so then we have our receptors and joint capsules, and these are going to be our free nerve endings that will detect these kinds of tensions and pressures within the joint. Chemoreceptors, as we've said, now I'm going to concentrate on the ones in the general senses. These are going to be responding to lipid-soluble, water-soluble substances in the surrounding fluid. And they have peripheral adaptation, but can also have central adaptation. These are going to be responsive to things like dissolved concentrations of carbon dioxide and hydrogen ion. We're going to find them in specific places in the body. So, for example, there are two places that are very important. The carotid bodies, which are basically right near the bifurcation of the common carotid artery into the internal and external carotid arteries. So the carotid artery services the area of the head. The internal carotid arteries will service the brain, and the external carotid arteries will come and service external structures of the head. And so this is going to give us information about carbon dioxide levels, which is going to be very important because we want to know how much oxygen is getting to the brain or how much carbon dioxide we have. So if we don't have enough oxygen getting to the brain, we need to do something about that. We also have the aortic bodies, and these are going to be on major branches of the aortic arch, which is the major vessel coming off the heart. So you're going to have your ascending aorta, which will become the aortic arch. You're going to have several blood vessels branch off of it, like the brachiocephalic artery, and you're going to have your left subclavian artery, which we'll learn about next semester. But basically, you're going to have these aortic bodies between these major branches that are going, again, to detect the amounts of carbon dioxide, oxygen, these kinds of things in arterial blood. Now, let's stop here, and when we resume next time, we will talk about the sensory pathways.